Question number 1. DNA contains some palindromic sequence which mark the site for the formation of replication fork, direct DNA polymerase to turn back to replicate other strain, are recognized by restriction enzyme, are found only in bacterial DNA. Correct answer is option B. Are recognized by restriction enzyme. Question number 2. Introns in gene encode the amino acid which are removed during post-translation modification, encode signal sequence which are removed before secretion of the protein, are the non-coding sequence which are not translated, are the sequence that intervene between two genes. Correct answer is option C. Are the non-coding sequence which are not translated. Question number 3. All of the following statements about post-transcriptional processing of tRNA are true except introns of some tRNA precursor are removed, CCA is added at 3 days and 7-methyl-guanosine triphosphate cap is added 5 days and some base are methylated. Correct answer is option C. 7-methyl-guanosine triphosphate cap is added at 5 days and ये ऑप्शन गलत थी. ये हमें क्वेश्चन क्या पूछा है कि ट्रू स्टेटमेंट पूछा है किसके बारे में, प्रोसेसिंग ऑफ टीआरएनए के बारे में. Question 4. Alpha amytin inhibits DNA polymerase 2 of prokaryotes, DNA polymerase alpha of eukaryotes, RNA polymerase 2 of eukaryotes, RNA dependent DNA polymerase. Correct answer is option C. Alpha amytin inhibits RNA polymerase 2 of eukaryotes. Question 5. Ciprofloxacin inhibits the synthesis of DNA in prokaryotes, DNA in prokaryote and eukaryote, RNA in prokaryotes, RNA in prokaryotes and eukaryotes. Correct answer is option C. DNA in prokaryotes. Question 6. All of the following statements are bacterial promoters are true except they are smaller than eukaryotic promoters. They have two co-sequence upstream from the transcriptional start site. Tata box is the site for attachment of RNA polymerase. Tata box has a high melting temperature. Correct answer is option D. Tata box has a high melting temperature. Yeah, it is the false statement about bacterial promoter. Question number 7. All of the following statements about eukaryotic promoter are true except they may be located upstream or downstream of the structural gene. They have two co-sequence. One co-sequence bind the RNA polymerase. Mutation in promoter region can decrease the efficiency of transcription of the structural gene. Correct answer is option A. They may be located upstream and downstream from the structural gene. Question number 8. In Sanger's method of DNA sequence determination, DNA synthesis is stopped by using 1,2-deoxyribonucleoside triphosphate, 2,3-deoxyribonucleoside triphosphate, 2,4-deoxyribonucleoside triphosphate, 2,5-deoxyribonucleoside triphosphate. Correct answer is option B. 2,3-deoxyribonucleoside triphosphate. Question number 9. tRNA genes have upstream promoter, downstream promoter, intragenic promoter, no promoter. Correct answer is option C. Intragenic promoter. Question number 10. All of the following statements about tRNA are true except it is synthesized as a large precursor, it is processed in the nucleus, it has no codon or anticodon, gene for rRNA are present in single copies. Correct answer is option D. ऑप्शन डी tRNA के लिए ट्रू नहीं है, बाकी सब स्टेटमेंट टीआरएनए के लिए ट्रू है। एंड क्वेश्चन 11, एंटीकोडन्स आर प्रेजेंट ऑन mRNA, rRNA, tRNA, hnRNA. Correct answer is option C, anticodones present on tRNA. Question number 12, codons are present on rRNA, hnRNA, tRNA, rRNA. करेक्ट आंसर इज ऑप्शन बी, hnRNA. hnRNA क्या है? अनप्रोसेस फॉर्म है किसका? mRNA का, इसलिए यहाँ पे आंसर में hnRNA है। क्वेश्चन नंबर 13, nonsense codon are present on mRNA, tRNA, rRNA, none of this. Correct answer is option A, nonsense codon are present on mRNA. 14, genetic code is said to be degenerate because it can undergo mutation. A large portion of DNA is non-coding. One codon can code for more than one amino acid. More than codons can code for the same amino acid. Correct answer is option B. More than one codon can code for the same amino acid. 15. All of the following statements about genetic code are correct except it is a degenerated, it is ambiguous, it is nearly universal, it is overlapping. Correct answer is option D, it is overlapping. ये वाला आंसर जेनेटिक कोड के बारे में करेक्ट है नहीं। ये सारे जो ऑप्शन है वो जेनेटिक कोड के लिए ट्रू स्टेटमेंट है। 16, all of the following statement about nonsense codon are true except they do not code for amino acid, they act as a chain termination signal, they are identical in nuclear and mitochondrial DNA, they have no complementary anticodons. Correct answer is option C, they are identical in nuclear and mitochondrial DNA. 17, a polycystonic mRNA can be seen in prokaryotes, eukaryotes, mitochondria, all of these. Correct answer is option A, seen in prokaryotes. 18, non-coding sequences are present in the gene of bacteria, virus, eukaryotes, all of these. Correct answer is option C, in eukaryotes. 19, non-coding sequence in a gene are known as cystone, nonsense codon, introns, exon. Correct answer is option C, introns. 20, splice site are present in eukaryotic mRNA, eukaryotic hnRNA, all of this. Correct answer is in eukaryotic hnRNA. 21, the common feature of introns include all the following except the base sequence begin with GU, the base sequence end with AG, the terminal AG sequence is preceded by a purine-rich tract of 10 nucleotides, an adenosine residue in branch site participate in splicing. Correct answer is option C. The terminal AG sequence is preceded by a purine-rich tract of 10 nucleotides. 22, a splice some contain all of the following except hnRNA, snRNA, some proteins, ribosome. Correct answer is option D, ribosome. 23, self-splicing can occur in some precursor of rRNA, some precursor of tRNA, hnRNA, none of these. Correct answer is option A, some precursor of rRNA. 24, Bribnov box is present in prokaryotic promoters, eukaryotic promoters, both A and B, none of these. Correct answer is option A, prokaryotic promoter. 25, Hogness box is present in prokaryotic promoter, eukaryotic promoter, both A and B, none of these. Correct answer is option B, eukaryotic promoter. 26, CAAT box is present in prokaryotic promoter 10 base pair upstream of transcriptional start site. Prokaryotic promoter 35 base pair upstream of transcriptional start site. Eukaryotic promoter 25 base pair upstream of transcriptional start site. Eukaryotic promoter 70 to 80 base pair upstream of transcriptional site. Correct answer is option D. CAAT box present in eukaryotic promoter 17 to 18 base pair. 27. Eukaryotic promoter contain data box 25 base pair upstream of transcriptional site cat. CAAT box 70-80 base pair upstream of transcriptional site both A and B, none of these. Correct answer is option C, both A and B. 28. All of the following statements about tRNA are correct except a given tRNA can be charged with only one particular amino acid. The amino acid is recognized by the anticodon of tRNA. The amino acid is attached to end of tRNA. The anticodon of tRNA finds the complementary codon of an mRNA. Correct answer is option B. The amino acid is recognized by the anticodon of tRNA. 29. All of the following statements about charging of tRNA are correct except it is catalyzed by amino acid tRNA synthesis. ATP is converted into ADP and PI in this reaction. The enzyme recognizes the tRNA and amino acid. There is a separate enzyme for each tRNA. Correct answer is option B. ATP is converted into ADP and PI in its reaction. 30. All of the following statements about recognition of codon on mRNA by an anticodon on tRNA are correct except the recognition of third base of codon is not very precise. Imprecise recognition of the third base results in wobble. Wobble is partially responsible for degeneration of genetic code. Wobble results in incorporation of incorrect amino acid in the protein. Correct answer is option D.